Hello, this is Ivan Rich. I'm CEO of HemoGenix. This presentation was given at a meeting organized by the German Institute for Standards on January the 20th, 2016 in Regensburg, Germany, on new test methods for cell-based products in cellular and gene therapy, initiatives for new standards. HemoGenix is one of the few companies that advocates the importance of metrology, or the science of measurement and validation, for the advancement of cell therapy and regenerative medicine products. Now, I do not profess to be an expert in metrology. There are organizations such as the National Institute of Standards and Technology, or NIST, who are the experts. However, it should be obvious that if you measure something, regardless of what it is, you should have the trust to know that what you're measuring is correct. After all, we trust that when we fill up our car, we are putting in a specific number of liters or gallons for which we have to pay a certain amount. Well, the same is true for any scientific measurement that we perform in the laboratory. And if that measurement cannot be trusted, then we have no basis for trusting our results or the interpretation and conclusions of those results. Cellular therapy and regenerative medicine are facing these problems, and unfortunately, many in the field think that they can trust their results explicitly because the assays they've been using have been used for decades. Nothing could be further from the truth. Now let's take a look at some of the background for metrology and validation in cell therapy. In the 1990s, the US Food and Drug Administration, the FDA, initiated an approach to regulate human cellular and tissue-based products. It might be expected that if something is regulated, then some form of measurement is required. In 2005, the Stem Cell Therapeutic and Research Act became law. It's just been reauthorized. This calls for high-quality umbilical cord blood units to be collected and stored. It requires that the stem cells are of high quality. Since the law requires that cord blood stem cells be of high quality, what does high quality mean? And how can it be determined such that the patient always receives a high-quality product? In 2009, cord blood was designated as a drug by the FDA because it resulted in systemic effects to the body. In 2011, the FDA published its guidelines on potency testing for cell therapy products. Autologous stem cell transplantation will become regulated in the future. There's no doubt about that. It applies not only to hematopoietic cells, blood cells, but also to every type of cell and regenerative medicine products. In contrast to the pharmaceutical industry, where metrology and assay validation is the norm, the cellular therapeutic and regenerative medicine fields are extremely lax and seriously lacking the basic understanding of what it means to measure and validate the results they obtain with the assays they use at the present time. This is not helped by the fact that regulatory agencies are often influenced by academia, resisting any change that would help improve safety for the patient, or contradictory guidelines published by the regulatory agencies themselves. In any event, there is a dire need for all results produced to make a decision for patient care to be trusted. At the present time, in the cord blood and stem cell transplantation industry, that is far from the case. Now consider these numbers. Umbilical cord blood is now used in over 100 different applications. In the United States, more than 210,000 umbilical cord blood units have been collected by public cord blood banks. Worldwide, about 750,000 have been collected. More than 5,000 cord blood transplants have been performed in the US, and worldwide, about 35,000. These and many other facts are used by both the cord blood community and the field of stem cell transplantation to demonstrate the success of what has been done and what is being done currently. Unfortunately, it belies a far more important fact, and that is, not a single cord blood unit collected and or used to treat patients by transplantation has ever been tested to ensure the presence of stem cells, let alone their quality and potency. As a result, 1 in 5 patients, which is about 24%, has succumbed to graft failure and died. The cord blood community, and especially the Cord Blood Association and the National Marrow Donor Program, or NMDP, might argue that the graft failure rate has been reduced. This would not be due to better testing, because they have prevented every possibility of doing this, but rather to the better treatment of patients after the damage has been done. So why is this the case? Since the 1970s, transplant centers and cord blood banks worldwide have relied on the total nucleated cell, or TNC count, using a cell fraction that contains cell impurities that mask and dilute the very small number of stem cells responsible for engraftment. A exclusion viability test that can produce erroneous results, CD34 membrane expression content that detects mature rather than primitive stem cells, and primary primitive non-stem cell progenitor cells. This gives a false indication of the stem cell content present. It also does not measure biological activity. And finally, the colony forming unit, or CFU assay, that is not only inaccurate and insensitive, but based on a TNC count, which cannot be used to measure stem cell growth. In fact, in many cases, transplant physicians do not even want cord blood banks to provide any other information other than the TNC count, because they do not consider the results from the other tests to be worth the money to perform. Yet, as we shall see, the use of TNC counts is also false. Everyone in the industry has been led to believe that the TNC counts are the beginning and end of any measurement needed, when in fact it is probably the worst measurement that could be performed and used as a basis to treat a patient. These 4 tests are the basis of hematopoietic stem cell therapy testing, yet they do not measure any stem cell property or characteristic that would allow cord blood banks and transplant physicians to guarantee the quality or potency of their products. So how can the situation be improved for cell and gene therapy products and to safeguard patients? Well, the answer lies in the implementation of measurement assurance. What is measurement assurance? It's knowing the level of confidence you have in the data that you are using to make a decision. It's having the data that provides credibility for the measurement result. In general, reagents or assay kits will be purchased from a vendor, and it is assumed that they will provide the intended results. In fact, there are hundreds of different aspects that can derail this assumption. The only way you can be assured that the results can be trusted is if measurement assurance is implemented for the test or assay to measure a specific outcome. When you purchase a reagent or assay kit, that is not the end of the story. It's actually just the beginning. So this diagram was provided by Dr. Anne Plant at the National Institute of Standards and Technology, NIST, near Washington, D.C. It shows that there are 3 steps in writing proper standards. The first is the test or assay itself that is used for measurement. However, the credibility and confidence of the assay and its use for providing data that might influence a decision to use a cell therapy product in a patient is based entirely on the reference materials, standards, and controls used to achieve measurement assurance. Without measurement assurance, the test or assay is useless. But only with measurement assurance can proper standard operating procedures, guidelines, and national and international standards be written. There is no doubt that implementing measurement assurance is probably the most mundane part of any test, but it's necessary. Very few assays can be purchased that incorporate measurement assurance. At HemoGenix, we believe that measurement assurance is a prerequisite for an accurate and reliable assay or test. Part of measurement assurance is the standardization of the assay prior to measuring any samples, to ensure that the assay is working correctly and that decisions can be made regarding the results. Some think that assay standardization is simply obtaining a reagent or assay that is quality controlled. In fact, assay standardization can only be accomplished by incorporating standards and controls. Without a standardized assay, it cannot be validated to achieve the required measurement assurance. So when you purchase or develop an assay or test, you need to ask some very fundamental but important questions. First, what are the target cells? In this particular case, they're primary hematopoietic stem cells, such as those used or those present in cord blood. Second, what's the goal or the intended use of the assay? In this case, it's to measure stem cell quality and potency. What's the biological process to be measured? Well, as we'll see, it's stem cell proliferation ability and potential. I'll come to that in a moment. What is the endpoint to be measured? Well, the endpoint is, in our case, intracellular adenosine triphosphate or ATP concentrations. And finally, what's the readout of that endpoint? The readout is actually bioluminescence. Now, the biological process to be measured is actually proliferation. You may have seen this diagram in other HemoGenix videos on YouTube. On the left side are the tests presently used in hematopoietic cell therapy, and I've mentioned those in a previous slide. You will notice that none of the tests, with the exception of HALO, and I'll come to that in a moment, actually measures cells in the stem cell compartment. For the present discussion, we're only interested in what happens at the stem cell level. On the right-hand side, you can see several stem cell properties and characteristics. It's important to notice that stem cell self-renewal, stem cell primitiveness or stemness, and stem cell potency all correlate with the proliferation parameter of capacity or potential. This in turn is indirectly related to proliferation ability. So how are these parameters defined? First, we have stem cell quality. This is the amount of stem cell proliferation at a specific cell dose and point in time. It is also the proliferation ability of the stem cells. Stem cell proliferation potential is the measurement of stem cell primitiveness or stemness. The greater the proliferation potential, the more primitive the cells, the greater their potency, and the higher the probability of engraftment. And finally, stem cell potency. This is the quantitative and validated measurement of biological activity of the active stem cell components that will predict an intended response, in this case, stem cell engraftment after transplantation. So how are these parameters measured? Well, one thing is certain. They cannot be measured with either CD34 or the CFU assay because neither of these assays measures proliferation. They can only be measured using an assay we've developed at HemoGenix called HALO. HALO is a standardized and validated assay for stem cell proliferation. It uses the most sensitive non-radioactive readout available, namely the measurement of intracellular ATP as a biochemical marker. All cells must produce ATP if they are alive and therefore viable. However, induced changes in intracellular ATP correlate not only directly with proliferation or cytotoxicity, but also cell number. In other words, when we stimulate the stem cells, there is a proportional increase in the intracellular ATP concentration. After incubation, the intracellular ATP is released by lysis of the cells. The ATP, as a limiting substrate, reacts with a luciferin-luciferase reagent to produce bioluminescence, which is measured as light in a luminescence plate reader. The output of the instrument is in relative luminescent units, or RLUs, and we'll come to that a little bit later. Now let's take a look at what HALO can actually do. The following slides all use cord blood as samples. As mentioned previously, all cord blood measurements are based on the TNC count. Everyone uses dye exclusion viability, for example, using trypan blue, or in this particular case, 7-AAD. And there is a good chance that the viabilities may be above 85%. But how do you know that this is the correct viability? How do you know that the stem cells being transplanted have the same viability as the TNC? If we now measure proliferation ability at 4,000 cells per well of the primitive hematopoietic stem cells called CFU-GEMM and the even more primitive lymphohematopoietic stem cells designated HPP-SP, then you can see that the metabolic viability of these cells is extremely low, and in fact many of the samples were actually metabolically dead. From these samples, the cells were further purified to a mononuclear cell, or MNC fraction. Here virtually all the samples produced 100% dye exclusion viability, but the stem cell viability and cell proliferation ability was extremely varied. In other words, dye exclusion viability of either the TNC or MNC fractions cannot be used to measure or even assume to provide accurate results regarding stem cell viability. Dye exclusion viability does not correlate with metabolic viability. Now let's look at another example. This example shows how proliferation potential is measured. However, it also again demonstrates the difference between the TNC and MNC fractions for each of the stem cell populations. If a simple 3-point cell dose response is performed using HALO, the slope of the linear regression is a measure of proliferation potential. You can see that the slopes using the TNC are much shallower than those for the stem cells from the MNC fraction. The steeper the slope, the greater the proliferation potential, and the greater the potency. In other words, the TNC fraction cannot be used for any stem cell assay. In fact, it cannot even be used to accurately measure progenitor cells such as BFUE or GM-CFC because the counts are so low. Rather than improving the situation to benefit the patient, cord blood leaders have decided to let sleeping dogs lie. However, this type of determination is the first step towards a potency assay. In contrast to what the majority of people in the cord blood industry have been told, a potency assay cannot be performed using a single point, and it must be performed using a reference standard. Again, none of the current tests used are actually potency assays. So a potency assay must measure the active components, quantitatively measure biological activity, predict the intended response, release the product for use, include a reference standard to determine the potency ratio, and include standards and controls for validation. And here you see how potency is measured for 32 cord blood segment samples. This procedure has been described in detail in another set of videos, and it's also been published. Using the previous method, all cord blood samples were compared to that of a cord blood MNC reference standard, and the measurement of potency, the potency ratio, calculated. The potency ratio is calculated from the slope of the stem cell dose response divided by the slope of the dose response for the reference standard for each of the stem cell populations being measured. The potency of the reference standard is always 1. Here you can see that many of the samples exhibit a potency ratio similar to or greater than 1. That's good. However, what is more important are the samples with a potency ratio less than 1. And of the 32 segments tested, 8 or 25% exhibited potency ratios where both stem cell populations were below the reference standard, indicating that their use in a patient would be undesirable. This result is very similar to the 24% graft failure rate after cord blood transplantation found in the literature. Now the question you must ask is, are these results credible? Can you trust these results? And the only way you can trust the credibility of the results is if you validate the assay. Unfortunately, virtually everyone in the cellular therapeutic field thinks they can validate an assay just by comparing it to an established assay. This procedure is actually called assay verification. For example, since HALO was originally derived from the traditional CFU assay, we can compare the results of HALO to 2 other methylcellulose-based colony-forming assays, namely Cameo4 and Cameo96 assays. And the results, as you see here in this 3-dimensional graph, will correlate. Similarly, we can compare 2 metabolic viability assays, such as the absorbance of MTS or MTT assay, with the ATP bioluminescence assay, and again the results will correlate. But this is not assay validation. Assay validation is providing documented evidence that the method does what it is intended to do. So now let's go back to the slide that I showed previously. The difference between this slide and the one I showed is that this one that is in this slide, the instrument is calibrated using calibration controls and standardized using a standard curve. So this is an example of the calibration and standardization of the assay. It is in fact the crux of the assay, because without it, you cannot say for sure that your results can be used. The red points show the low, high, and extra high calibration controls. The output of the instrument, as I mentioned previously, is in relative luminescent units, or RLUs. With the standard curve, the non-standardized RLU values can be interpolated into standardized ATP concentrations. By performing a standard curve every time samples are measured, you can compare the results over time and between different conditions. Now let's take a look and see how all this comes together. First, we have to ensure that the accuracy of the ATP standard curve is within proper ranges. The standard has to be diluted to perform the ATP standard curve. Therefore, we can compare the expected concentrations with those that we actually obtain. As you see, the results we obtain are extremely close to the expected values. The important parameter here is the percent coefficient of variation, which must be 15% or less. As you see, we are within range. This slide shows the parameters of the standard curve itself. And we're looking at up to 16 different dose response curves. In this case, the slope is the important result, because this too must be within a certain range. Here you can see that the slope is 0.973 and the percent CV is 1.233. You'll see shortly how measurement assurance is implemented using this type of data. Now we can start looking at the accuracy, sensitivity, and selectivity of the assay results for the two stem cell populations. Receiver operator characteristics, or ROC curves, allow this to be performed. It's done by comparing the background or unstimulated cells with cells that are stimulated to detect the stem cell populations of choice. Now the plot demonstrates what's called true positive rate or sensitivity against the false positive rate or specificity. There's usually a trade-off between sensitivity and specificity, so if one increases, the other will decrease. The closer the curve follows the left axis and the top border, the more accurate the assay. A 45-degree diagonal indicates that the assay is actually worthless. The area under the curve, or AUC, is a measure of accuracy. This must be between 0.5 and 1, with 0.5 being a worthless assay and 1 being an ideal assay. No assay is ideal. The area under the CFC-GEMM curve is 0.75, while that of the HPP-SP is 0.73, with a high probability of accuracy. Just for comparison, the CFU assay for these two stem cell populations is close to the diagonal, and therefore the accuracy, sensitivity, and selectivity is virtually worthless. Now let's take a look at precision, which is the measure of reliability and reproducibility of the assay. There are two types of precision measurement. The first, seen here, is within-run intra-batch precision. Here we're looking at a cell dose response for the two stem cell populations in the background. The aim is to obtain CVs of 15% or less for the higher cell doses and 20% or less for the lower cell dose. This is called the lowest level of quantitation, or LLOQ. As you can see, there are some outliers shown in red, but the values are otherwise within the required range. The second type of precision is between-run interbatch precision, and here you can see that all of the values are again within the required range for the CVs. In other words, 15% or less. Now, one of the last parameters to be measured is robustness. There is intra and inter-laboratory robustness. Robustness is how well the assay can withstand changes, the most important of which is whether the assay can be transferred from one lab to another and still get the same results. These results were published in 2008 and show the interlaboratory robustness between the same core blood samples tested at HemoGenix and at the Puget Sound Blood Center in Seattle. As you can see, there is a high degree of correlation, with a correlation coefficient of 0.94, indicating that that halo can be transferred from one lab to another and produce similar results. This is certainly not the case with the CFU assay. So based on the validation parameters, it's possible to provide the HALO measurement assurance characteristics. The first 5 parameters must be attained to continue to measure samples, and failure to achieve these parameters means that the assay is neither standardized nor validated, and the results obtained will not provide the confidence required. So the first parameter is the linear regression coefficient, correlation coefficient, which must be greater than 0.997. The most important is the linear regression slope, which is, in this particular case, is 0.937. And we provide a slope range allowed, which is between 0.796 and 1.07. This is where the 15% range comes in. There's a low calibrator, high calibrator, and an extra high calibrator. And all of these have an allowable range. Parameters 6 and 7 provide the information required for cell viability. So parameter 6 is the lowest ATP value indicating unsustainable proliferation, which is 0.04 micromolar of ATP plus minus 15%. The ATP value below which cells are metabolically dead is 0.01 micromolar of ATP. Parameter 8, which is assay linearity, which in this particular case is greater than 4 logs, is required by all biological assays. They must demonstrate linearity over a specific range. And parameters 9 to 11 provide the limits of the assay, which in this case is between 1,000 and over 25,000 cells. The assay sensitivity is 0.001 micromolar of ATP, or 1 log lower than the ATP value for metabolically dead cells. And the assay sensitivity is between 20 and 25 cells. So here we see the stem cell assay validation. The accuracy, sensitivity, and specificity will depend upon what is being measured and whether the results provide the intended goal. These values will be different for different assays. However, in general, the accuracy, or the percent of correct outcomes, is approximately 95%. The receiver operator characteristics, as I've mentioned, are 0.73 to 0.752. The precision is equal to or less than 15%, with an LLQ of 20% or less. Robustness, the intra and inter-laboratory robustness, is also approximately 95%. And these assays, or many of these assays, have high throughput capability, and we can measure that with a so-called Z factor, which is greater than 0.76. So what is the goal? The goal is to ensure that all patients receive the highest quality and most potent cell or gene therapy product available to effect a cure. Patients are being betrayed into thinking that they are receiving the best quality product. And it is up to academia, industry, regulatory agencies, and standard organizations to ensure that all cell therapy procedures and assays are safe and meet patients' expectations. And finally, the stem cell transplantation community thinks that once they have a test or assay, that is the beginning and end of what they need to do. Without measurement assurance, no one has any confidence in the results they generate. Regardless of whether the cell source is autologous or allogeneic, umbilical cord blood, mobilized peripheral blood, or bone marrow. All of the tests currently used are based on false assumptions and very serious defects. The stem cell transplantation community does not want to change or improve their testing procedures, because standard operating procedures, or SOPs, also have to be changed. Unfortunately, SOPs are the number one priority, not really the patient. And the stem cell transplantation community have yet to understand that costs could be reduced and patient safety and clinical outcomes could be significantly improved if measurement assurance and best practices were implemented. So if you're part of the community and involved or responsible for the production of cell therapy products, and or make a decision as to whether the product can be used, then remember that if you were a patient, you would want the highest quality product yourself. If you're a member of the public, patients thinking about storing umbilical cord blood, or a patient, ask the cord blood bank or the transplant physician whether they have the confidence to guarantee that the product has the highest quality and potency to be used to produce a successful outcome when the need arises. If they say yes, don't believe them. Get them to show you the evidence of measurement assurance that they have the confidence to guarantee their product. Thank you, and I hope you've enjoyed this presentation. Goodbye.